The problem isn't just supply chains. Heat is breaking the enzyme that makes most of our food. Saturday at 4:40, a parent holds flour beside a supermarket receipt. Phone calculator glowing against warm window glass. They cross flatbreads from the week's meals and plan thinner portions before Monday's rent. The shelves are full. Their bag is not. Food prices now stand at their highest since January 2023, as extreme heat scorches major crop regions. In July 2026, wheat jumped 5.8%. Before any field looked empty, the shock reached dinner. Since 2021, rising temperatures and associated impacts accounted for 60% of the £605 average rise in UK household food bills. Heat and drought account for more than half of annual crop losses worldwide. What fails inside a leaf before harvest disappears? In laboratories, researchers compare crop proteins with enzymes from heat-loving algae found around volcanic springs. AI predicts shapes experiments cannot easily capture. Gene editors build candidates into plants. The goal is to locate the moving parts that buckle under heat. Then test whether biology can be made less fragile. The race comes down to this. Can scientists protect photosynthesis in real heat, then deliver the result safely and affordably before hotter harvests rewrite the next receipt? Inside each leaf, a molecular gate takes carbon dioxide from air and turns it toward grain. The gatekeeper is rubisco, Earth's most abundant protein, roughly 0.7 gigatons, and up to half the protein in some leaves. It supports virtually all life. But abundant does not mean optimized. Rubisco evolved in an atmosphere richer in carbon dioxide and poorer in oxygen. Today, this mediocre catalyst often grabs oxygen instead. The error triggers photorespiration, a cleanup cycle consuming 30% to 40% of a C3 leaf's energy, and releasing 25% to 30% of carbon already fixed. Energy that might fill grain simply circles backward. Yet the hardest failure is not rubisco itself. Rubisco activase, or RCA, is the chaperone that pulls inhibitory sugar phosphates from rubisco's active site. In most plants, RCA loses function between 35 and 40 degrees Celsius. Heat mainly disables this helper rather than destroying rubisco, causing photosynthesis to shut down during heat waves. Scientists are attacking that failure. AlphaFold predicted unavailable shapes of photosynthetic enzymes, including glycerate kinase, or GLYK. In its plant form, researchers found 3 flexible loops that wobble out of shape under heat. Hot spring algae reveal structures that remain stable. Generative models propose rubisco variants for greater thermal stability and catalytic activity. Predictive systems combine genomes, phenomics, and climate data to model stress tolerance in future weather. This computational genomics can compress breeding cycles from decades toward years. Candidates still require physical proof. Multiplex CRISPR-Cas9 has removed native rubisco genes from tobacco, creating a clean slate for testing engineered versions. Another clue comes from salt-tolerant plants. Overexpressing one RCA gene raised photosynthetic rates 41% under stress. This is not a digital crop. It is a sequence of predictions, edits, growth chambers, and failed plants, searching for one that keeps filling grain under heat. But greenhouse success is not cheaper bread. Altered genes can cross-pollinate, reshape ecosystems, or affect soil microbiomes. That uncertainty justifies field trials. Approval, especially in Europe, can take years or decades. Seeds must then be multiplied and distributed. If patents concentrate ownership, farmers facing the harshest heat may pay first and benefit last. The trade-off is real. Rapid access versus evidence that release is safe. In 5 years, field trials can test engineered proteins under real heat. In 10, successful traits may enter regional seed systems. By 15, tougher enzymes, field phenotyping, agronomy, and fair access could protect harvests unless heat outruns deployment. Back in the kitchen, the calculator still cannot discount a lab result. That parent's bill changes only when resilient seeds are proven, approved, shared, and affordable. Can scientists raise yields in farm heat without ecological harm? Were seed systems too slow and costly to reach families first? CivilizationX will track the answer. The constraint is real. The data exists. Whether the systems built around it can act on it at scale, that is the next engineering problem.